Now, this protein portion of the macromolecules or carbon and its molecules, uh, protein accounts for 50% of the dry mass of most of the cells. And like I told you earlier, protein is a machine in your system. It's like having a car and gas. Protein is the car, carbohydrate is the gas. So if there's no protein, there's no function that happens in your body. That is why when you don't have lactase, even though you can have ATP, if you don't have lactase, you cannot break down lactose. So you drink milk, you have challenge digesting it because there's no lactase in your system, and lactase is a protein because most enzymes are proteins. All right. So protein function in defense mechanism. They do storage, transport, cellular communication, movement, and structural support. So these are characteristics of proteins. I mean, these are functions, characteristic functions of protein. You see some proteins that does enzyme activities, and those proteins, the enzymatic activity, you can see that here, they help to accelerate reactions. They help to speed up chemical reactions. They act as enzyme, bringing up chemical reactions together. That is the case of lactase that I just mentioned. If lactase is not present, lactose cannot be broken down into glucose and galactose. And in that case, lactose is going to create a nuisance in the body because it's there. The body is struggling to digest it. The body cannot digest it because the guy that does the digestion, that hates the digestion, is not present. And that is lactase. Okay, so when people have lactose intolerance, you see that they have diarrhea, they have bloated stomach because the lactose is there and it's making the stomach to be bloated because the body cannot digest it. All right. Now, protein can also act as storage for storage, can read that up, they can act as hormones, they can act as contractile and motor proteins. For some of you that will be taking hair and peel later, you will see, or some of you are taking it now, you will see when you are talking about chapter 9, when you're looking at contraction of your muscles, proteins help with contractions. So we have contractile protein, the tropomyosin, and troponin. Those are contractile proteins. They are regulatory proteins. We have the actin, the myosin. Those proteins help for contraction. So without protein, my muscle cannot contract, right? So we have defensive protein, defensin. We have proteins that are due for transportation. They help to transport molecules on the cell membrane. Like the cell membrane is selectively permeable. It does not allow everything to come in and out of the cell. So it's selective to what comes in and out of the cell. And so there are some proteins that help to carry some materials that are needed by the cell but cannot pass through the cell membrane. So think about it. Your cell needs glucose, but glucose are quite bigger and they cannot pass through the cell membrane by themselves. So they need protein as a transporter to transport them. Your cell needs some ions, like sodium ions. The cell needs it. They cannot pass through the cell membrane by themselves, so they need transporter to carry them. So we use sodium ion channel to carry such proteins. Then receptor proteins, they act as receptor on the cell membrane when there is when there are some hormones, they bind on that receptor, and then the protein can create a cascade effect or does some genetic some reactions that will cause the job of that protein as either a second messenger or so to be achieved. All right, then we have structural proteins, which are the guys that you see in your connective tissues predominantly. So you see collagen, elastin, all of those are structural protein. Like collagen now, for instance, your skin is going to be like this, but as we age, our skin begins to wrinkle. It begins to wrinkle. The reason the skin is wrinkling is simply because the collagen fiber is becoming depleted. The weaker the collagen, then the skin will not be able to retain its structure again. So those are structural proteins. Now, I will go ahead and talk about the types of proteins. All right. Now, then I will show all of this to you. These are proteins have monomers, and the monomers for protein are the amino acid. And so these are different amino acids, which are the monomer for protein. When I was going through carbohydrate, I told you the monomer for carbohydrate are the monosaccharides. The monomer for nucleic acid are the nucleotides. So the monomer for proteins are the amino acids. So different amino acids combine together to give us what we call proteins. So we have 4 types of proteins. We have the primary proteins. I'll talk about all of them here, and I'll show you the images. We have the primary proteins, the secondary proteins, the tertiary proteins, and the quaternary proteins. The primary proteins are what you see here. This is an example of primary proteins. These proteins, they are not coiled on each other. They are just chains of amino acids that are connected together by what we call peptide bond, which is what you see here. All right, there are sequence of amino acids that are connected by peptide bond here. When you have straight chain proteins like that, they are non-functional. They don't have functions as protein. They are functional in that they had to produce secondary protein and form the chain, but they cannot function as structural protein in our system. They cannot function as in digestion in our system. They cannot function like that. So we say that they are primary proteins. An example of that is just the way you see the polypeptide. It's just sequence of amino acid. Now we have the secondary proteins that we call alpha helix or beta sheet. Now, if you look at this is like primary proteins. These are primary proteins, but they have connections together now. They are becoming coiled on each other. They are becoming bonded. So it's an advanced level of proteins, but it's still not going to perform the function of a protein. Then we have this tertiary protein that is now coiled, twisted, and folded on each other. And now this is a tertiary protein. It has formed a 3-dimensional shape. Proteins are only functional when they have 3-dimensional shape. So this protein, for instance, now is going to be able to carry hemoglobin because it has a 3-dimensional shape. This is what we call functional protein. Tertiary and quaternary proteins are functional proteins. So this is a functional protein. Collagen is a 3-dimensional shape protein and it is functional. This is a functional protein. It is quaternary protein. It is 3-dimensional shape. It is functional. Now, if the difference between regular blood cell and sickle cell is simply because there is a problem with the structure of the protein different from what it is. This shape is what makes this protein to be able to carry 4 oxygens. But when there is sickle cell, there is a mutation. The mutation affects the shape of the protein. When the shape of the protein is affected, there will be oxygen deficiency. That's why it is called sickle cell anemia, because there is deficiency in oxygen supply due to protein, due to inability of the hemoglobin to carry oxygen because the structure of the protein has been affected. That is what is called sickle cell anemia. Now, this is what happened. So now if you look at this is the primary structure of the protein. This is the sickle cell. Instead of glutamine here, it has changed to valine. Now, this is the regular shape. This is the shape of the sickle cell. This is the shape, the regular hemoglobin that should carry oxygen, and oxygen will bind there. Now, these ones already are affected. Oxygen cannot carry, it can't carry oxygen. So now this is the shape, but this is this shape. Now, eventually, this is the red blood cell shape here. This is the shape here. This cannot carry oxygen the way this will carry oxygen. And that's why there will be oxygen deficiency. The oxygen deficiency is what leads to the crisis, is what leads to anemia, because there's oxygen deficiency. Now, what happens when temperature rises or when pH rises and they ask you to run to emergency? When temperature rises, they ask you to run to emergency because higher temperature, those proteins became functional by coiling together. They became functional by bonding together. They became functional by twisting on each other because of the bond. High temperature, high pH can break those bonds. When those bonds are broken, the protein will lose its shape. When a protein loses shape, the protein becomes denatured. So we will say the protein has lost its nature. When a protein loses its three-dimensional shape, that protein has lost its nature. When a protein loses its nature, it becomes denatured. So at that point, we say the protein has been denatured. A denatured protein cannot function. And that is why when temperature rises up to 104, because the doctors know that something may be at stake if temperature rises beyond that 104 and it keep increasing. And that's why they say run to emergency. The person can have seizure, the person can faint, some systems, some things can be affected, it can affect metabolic activities. And so that's why they say run to emergency when temperature rises beyond 104, because you may not be able to control what's going to happen after that. Now, that is that. All right. So, let me see. Okay, so I'll talk about nucleic acid there briefly on the part that is there. Now, for nucleic acid, the nucleic acid are actually the DNA and RNA. DNA is deoxyribonucleic acid, DNA, and RNA is ribonucleic acid. Now, the DNA is what is responsible for our genetic composition. RNA is derived from DNA. RNA is single-stranded, and DNA is double-stranded. DNA is what you see in the nucleus, which is what I'm showing you now. And it's going to make mRNA from there. And then mRNA is going to make proteins. This is what we call central dogma of biology. This is what we call gene expression. So the DNA provides direction for its own replication. The DNA directs synthesis of messenger RNA. And through messenger RNA, control the synthesis of proteins. Now, nucleic acid, what is the polymer? What is the monomer of nucleic acid? The monomer for nucleic acid are called nucleotides. Nucleotides. What makes up a nucleotide? A nucleotide is made up of, let me use my annotations or my annotation. A nucleotide is made up of, number one, nitrogenous base or nitrogenous bases, which will be adenine, guanine, cytosine, uracil, and thymine. RNA doesn't drink tea, so RNA doesn't have thymine. RNA doesn't like tea, RNA only likes you. So RNA have uracil, it doesn't have thymine. All right? And I'll show that to you. So those are nitrogenous bases. Adenine, you see it called A, thymine T, guanine G, cytosine C, and uracil U. Then it has pentose sugar. The pentose sugar is called ribose sugar or deoxyribose sugar. So here's the pentose sugar. The difference between ribose sugar and deoxyribose sugar is that deoxyribose lacks oxygen on one of the carbon here. And then there's the phosphate. So here is the nitrogenous base. Here are nitrogenous bases, nitrogenous bases, nitrogenous bases. Here are the phosphate. and here is the sugar. All right, so let's go. So the portion of nucleotide without the phosphate is called nucleoside. So if there's without phosphate is nucleoside, with phosphate, everything is called nucleotide. So here is the monomer for nucleic acid. Let's go. Now, so you see, here are the here are the nitrogenous bases. You have cytosine, you have thymine, it is T. I told you RNA doesn't drink T. RNA doesn't like T, RNA likes U. So the U is in RNA, only DNA drinks T. RNA doesn't like T, RNA only likes U. Okay? Now, so you have adenine. So we say that A pairs with T. So A will always pair with T, and C pairs with G. All right. So these three, we call them pyrimidines. These are the pyrimidines, and these are purines. So when you want to pair them, I'm going to use my test. So when you pair them, A pairs with T, and G pairs with C. DNA is double-stranded. But anywhere you see T in RNA, you replace it with U, because RNA doesn't like T, RNA only likes U. So each nitrogenous base has one or two rings. The pyrimidines have one ring. The purines have two rings. And the way to know the purines is, this is the way to know the purines. If there's a way you want to never forget the purines, obviously, when you see the structures, they have double rings. But they have 9. They are the one that ends with 9 or nin. Those are the purines. This is not nin, this is cytosine, this is thymine, but the purines are the one that ends with 9. Those are the purines, the adenine and the guanine. All right, let's go. So the sugar in DNA is deoxyribose. And think about it. What does deoxy mean? Deoxy means that on this position, look at it. In ribose, RNA have ribose. DNA have deoxyribose. That's why the name is ribonucleic acid, deoxyribonucleic acid. All right? So DNA, the name is deoxyribose, meaning that the sugar, the pentose sugar there, pentose simply means 5 carbon sugar. So both sugars are 5 carbon sugars. But here, when you look at the carbon 2, there's H there. But when you look at carbon 2 here, there's OH there. So it is deoxy. It has lost an oxygen. Deoxyribose sugar. The name of the sugar is ribose sugar, but now it has lost one oxygen, so it is deoxyribose sugar. All right? So adjacent nucleotides are joined by covalent bonds that form between the OH group of carbon 3, which is what is here. So the carbon 3 here is where they join together. Let me see if I can show you that here. All right, cool. So you see the carbon 3 here is the point where they join. So when you are looking at the DNA strand, it's either 5' to 3', 3' to 5' because of the carbon location. So there's going to be, you see, OH group is on the carbon 3, on the 5 carbon, and then phosphate is going to be on the carbon 5. So like what you're saying here. So you see, this is the phosphate joining the carbon 5 on the, this is the sugar. So this is going to be the point where it's going to attach to the nest, and the phosphate is going to be on the carbon 5. So carbon 5 is going to be from here. Then the 3 is where the OH is where the OH group is. So like what you see here. So you see the OH here, OH ends, phosphate is here, carbon 5. OH here, phosphate here. So like what you see here. Let me do my annotation. So you see here, You see, on carbon 5, phosphate joins carbon 5, and OH is going to be on carbon 3 here. If this has continued now, something is going to be here. So you see, carbon 3 here, whereas this is phosphate here, joining carbon 5. All right? All right, RNA is, so this linkage, these links create a backbone of sugar phosphate unit with a nitrogenous base appendage. So what you see as the main structure is if you look at this now, you see that the main backbone is sugar and phosphate. These are appendages, they are chain, they are attachments, the sugars, the nitrogenous bases, they are attachments. The main backbone of DNA is sugar and phosphate. Nitrogenous bases are the attachments. All right, so the sequence of bases along DNA or mRNA polymer is unique for each gene. All right, now if you look at this, RNA molecules that usually exist as single, I told you they are single-stranded, whereas DNA is usually double-stranded, and we call it double helix. We say it is a double helical structure. Then in the DNA double helix, the two backbone runs in opposite direction, which is what I was saying earlier. We say that they are anti-parallel. So if one runs 5 to 3 prime, the other is going to run 3 prime to 3 prime. So it's like this. One run 5 prime to 3 prime, the other run 3 prime to 5 prime. They are not, edges are not together. They are anti-parallel to each other. So it's going to be 3 prime to 5 prime, then 5 prime to 3 prime. That's the way to run. So one DNA molecules include many genes. All right. So nitrogenous base pair up and form hydrogen bonds. Again, adenine pairs with thymine, AT, and guanine pair with cytosine. This is called complementary base pairing. All right. Again, whenever you see an RNA, you will not see T there because RNA doesn't like T. RNA only likes you. All right. Here is the end of chapter 4. Please feel free to let me know if you have questions, and I will take it and respond to your questions. Thank you.